Wanted to cover a few things that I had heard that there was some confusion about. I found these other illustrations on some of my senior pre-med biology courses about urinary. So, what is this whole thing called? Renal corpuscle. The loops inside, the glomerulus. Notice the color of the afferent arteriole. Well, they got them opposite here from our book, but we're still coming in with the afferent, exiting with the efferent. But you notice they're pink. The yellow is illustrating what? Yes, the things on top of it. Collectively, what do we call those things on top? The podocytes are the individual pieces, but collectively, the visceral layer of Bowman's capsule. This is the parietal layer of Bowman's capsule. Now, we're going to look closer, take a little inset. You can see the foot processes of these podocytes as they're wrapping around the capillary. This one even illustrated these holes inside the capillary. What are the holes? Fenestrations. Now, when we look at the filtration membrane, even closer, they've sort of exploded that view. Have you ever seen an exploded view of like a piece of equipment or a machine? So they've exploded this. The first filtration barrier, the fenestrated endothelial cells. Then this solid layer that they're kind of illustrating, it's not cells, but that's the basement membrane. And then here they're showing these slits. They're just lines in the illustration, but slits between the podocyte foot processes that are called pedicels. And you can see slit diaphragm. There's a membrane that goes across there. All right? So that is our filtration membrane, those three components. Now, if you were to look at this with an electron microscope, this is one of those things that sometimes when you look microscopically at biology, it looks really cool because it looks like that. That is an actual look. You're in Bowman's space. The parietal layer is over your head. You're just surrounded by it. You're looking down on one of the capillary loops of your glomerulus, and these are all the foot processes that wrap around that capillary loop to be your third filtration membrane. These are some of the major processes. The little processes are the pedestals or secondary processes, and there's the nucleus of the podocyte with all of these processes coming off of it. Does that help understand and see the filtration membrane? Okay. All right, this is why we've got to get to this today, because this may take a little bit more time and I want to spend more time on it. If we have time at the end, we can go over the quiz. Have any questions, we can do that. But we got to get through this. So countercurrent multiplier is going to be water reabsorption. How do we move water in the kidney? We can't push it, right? Can we actively transport water? Bless you. No, we can't actively transport it. The best we can do are put some of these water channels in places, and what do we call those water channels? Aquaporins. Are they active transport carriers? No. They're facilitated diffusion. So we have to create concentration gradients, not of water, but of solutes. And so this countercurrent multiplier method is going to be a means with which we produce a high concentration of salt in the area that we need water to move. And this area is going to be in the medullary spaces around the tubules of the nephron, particularly around the loop of Henle and around the collecting tubule. Now, in this illustration, we're going to spend a little bit of time here, but here's the cortex, that's the medulla. What would you call that dashed line that divides the cortex and the medulla? There's a specific term we use. corticomedullary junction, and if you live right here where that cortex word is, where do you live? In the juxtamedullary subdivision, because you're near the medulla.
If you live right above it?
Yeah, if you live like right there, you know, we got our nephrons and our glomerular corpuscles there. For those long nephrons, it's a juxtamedullary nephron. So that's going to be adjacent to the medulla. Okay?
So these are why only the juxtamedullary nephrons are really the ones that are responsible for water and salt.
Mostly responsible. Not the only ones, they're mostly. Right, and the reason is, as you go deeper into the medulla, the ionic nature of the interstitial spaces is getting more and more hypertonic. You're going to see a gradient of salt concentrations where salt gets more concentrated and more concentrated and more concentrated as you go down. This is because we are going to actively transport sodium, and chloride's going to passively follow throughout that medullary space. So as we first enter the medulla, you haven't had as much of these transport mechanisms working, so it's going to be the least hypertonic. But as we go deeper and deeper and deeper, we have had more of these transport channels able to influence how much salt is in these deeper and deeper regions. So when we start right here at that juxtamedullary region or the corticomedullary boundary, we're going to see that the tonicity of our filtrate is 300 milliosmol. Have we heard that number before?
Yes, sir.
300 milliosmol. Why does that sound familiar?
Because that's
That is the osmolality of your plasma of your blood. So our filtrate in the proximal convoluted tubule is what kind of tonic relative to the blood?
hypotonic. No. Oh,
If it's the same, it's isotonic. We're about to change it. Now, I said as we got deeper and deeper and deeper, it was going to become more hypertonic, right? In the interstitial space. So we're just working with interstitial space right now.
Okay.
The medulla.
Okay.
And when we get to the bottom of the loop, especially those juxtamedullary nephrons, the surrounding interstitial environment is going to be 1,200 milliosmol. You got a lot more solutes down here, down at the bottom. This is just not my day. All right, so we're going to go from 300 up here to 1,200, and there's going to be a gradual gradient going down. Does everyone understand that? When we move sodium and chloride, we're going to create a deeper and deeper and a bigger and bigger ionic concentration the deeper we go into the medulla.
So is it becoming saltier as you go down?
It's going saltier as we go down.
So water is coming out. Salt.
Not, we're not yet.
Okay.
Not yet. We're putting the Legos out before we build our Star Wars model. Okay? Now, two things I want you to understand in our illustration. This on the left, 300 milliosmol, proximal convoluted tubule. And you can see with this green arrow on the inside, see how it's going down? This is the flow of our filtrate. So this part of the intermediate loop, loop of Henle, nephron loop, whatever name you want to call it, this is called the descending limb because it's going down into the medulla. On this side, the filtrate is going to go up toward the distal convoluted tubule. That is going to be the ascending limb. So we're going to be using these terms. Now, notice right away, we start at 300 milliosmol in our filtrate. When we get to the distal convoluted tubule, it's 100 milliosmol. That's what relative to blood? Hypotonic. Is that going to be a problem? It's a trick question.
No.
Is that going to be a problem? No, because we're just about to, that's urine. Urine's going out of the body. It's not going back into the blood. All right? Speaking of blood, this in the red is our vasa rectae. These are the capillaries that are going to help with exchange in the loop of Henle. Notice the flow of blood relative to the flow of the filtrate. It's the opposite direction. So our descending limb of our vasa rectae, which is also some called the arterial rectae, it's coming down beside the ascending limb of the loop. And over here, vice versa, the ascending limb, or the vena rectae, is going up, and this is the descending rectae of the loop, or the descending limb of the loop, okay? So the flow of the vasa rectae is opposite that of the filtrate in the intermediate loop, okay? I mean, we're setting the stage for turning the switch on. So here we go. We're going to start over here. Ignore that part. The ascending limb of our loop is impermeable to water. Water will not go in or out of the ascending limb of the loop. But here where it says permeable to salts, that's kind of half-truth. It's permeable because we have salt pumps in the ascending limb. And what do we call the salt pumps? Sodium potassium pump. Actively transported out, chloride's going to passively follow, boom. We have salt in the interstitial spaces. And even though we're just looking at a picture of one, understand how many nephrons do we have in the kidneys? Millions. So we've got these loops on top of loops on top of loops all over. So we're pumping a lot of salt out into this interstitial space. And that salt is going to get more and more concentrated the deeper we go. From 300 here all the way down to 1200 or more as we get deep with our loop and our bend down there at the bottom. So salt is being cranked out of our ascending limb. But water can't follow because water just can't get in or out. But over here in the descending limb, this part doesn't have the salt pumps, but it is permeable to water. So the descending limb, water can move out into that hypertonic environment. In the ascending limb, salt is pumped out to make the environment hypertonic. Those are the two big pieces of this countercurrent multiplier. And this is how we are going to reabsorb constitutively, not regulated, about 25% of that 180 liters of water that we had in our filtrate.
That was my question, is it because it reabsorbs it at the beginning?
Well, we're going to reabsorb 65% of the water at the PCT. We're doing about 25 here. The last 10 is going to be with ADH and the aquaporins in the collecting tubule. So, as we put more and more salt out, that is going to make the environment more and more hypertonic. And as the filtrate comes into the loop of Henle, as soon as we hit those parts that are permeable to water, where is the water going to go? Out of the filtrate into the interstitial spaces, and what do we call that? Not filtration. It's diffusion. It's diffusion, but as far as a direction of movement, it's not filtration, it's absorption or reabsorption. We're getting it out of the filtrate and we're on the way to getting it back into the blood. But first, we're going to get it out of the filtrate.
So basically it gets all the salts out of the loop, so that way we can conserve water.
The whole point of doing the salt is to conserve the water. Okay? So, as soon as we get into this first part, water starts coming out, but the salt can't. So as water leaves, what happens to the osmolality of the filtrate? We're concentrating the salt. So we're going to go from 300 to 400 to 500, 6, 7, 8, 9, 10, 11, 12. By the time we get to the bottom of the loop, the concentration of salts in the filtrate is isotonic with the interstitial environment because you're pulling out the water and concentrating the salt in the filtrate, right? But do we want 1,200 milliosmolar fluid either going out in our urine or going back into our blood? No. How are we going to return this to its normal step? Because we can't get rid of the water over here to dilute it. I mean, to, dilute it.
Pull the salts out.
You pull the salts out. You don't have to add water to dilute it. What we're going to do is remove the salts. So now in the ascending limb, as we go back up, we start pulling out more and more and more and more of the salt. And actually, by the time you get to the distal convoluted tubule, we pull out so much salt because we want to resorb the sodium chloride. We're all the way down to 100 milliosmolar.
So do we have more water by the end of this whole thing? Is there more water in?
We're keeping the water. Remember, how much urine do we make a day? Maybe a liter. We got to make 300 to 500, period. 800 to 1,000, milliliters, so a liter a day. But we have 180 liters of filtrate we got to keep. This is one way we're keeping 25% of that.
It's going to the interstitial tissues?
Interstitial tissues, we got one more step, okay? But we got it out of the filtrate. We're keeping it from becoming urine.
It's going in the body, but you're not peeing it out.
Yes. Okay,
Yeah.
okay,
So we
okay,
haven't got it back
okay.
into the blood yet. That's the next step.
Okay.
Okay.
It's just floating in the river with you. It's not in the life.
Yeah.
How fast is it going?
How fast is it going? That's going to be your glomerular filtration rate. And we haven't calculated that, and we're not going to. But again, if you make 180 liters a day, then you're resorbing all but 1 liter a day. So it's going about as fast as you make it. Yeah.
So right now our filtrate is real salty, right?
Our filtrate's going to be really, really salty down here at the bottom. But by the time it gets up here, it's not as salty as your blood
Okay.
because we keep the salt.
Okay, and the water's in the interstitial.
Correct. The water is in this white space right here. Okay? The water is in between the loop and your vasa rectae. We haven't got it into the blood yet.
So if there's hardly any water left, what's left? In the
Well, again, you're going to make maybe a liter of water a day. So there's fluid still there. And to get the waste out, you've got to make about that 500 mils of urine a day. So that's the minimum fluid volume you need to keep the kidneys working and to clear everything. Okay? All right. So this summarizes what we've been talking about. Over here, this is a collecting tubule, but here is our interstitial osmolality. And see how the deeper you go in the medulla, you get more hypertonic? Here is our loop. This is our descending limb. See, we start at 300, 4, 6, 9. Why did they skip 8? Drives me crazy. 12. And notice we're here at 12. Well, the interstitial's at 12. But we go back up, and this is where we're actively transporting the sodium out. Chloride follows. Water is leaving here, and because the water leaves, that's why your filtrate becomes more hypertonic too. But when we come up and get rid of the salt, that's what's making it return to isotonic and even go hypotonic in this area. All right?
Urea is nitrogenous.
I almost blocked that out. I was like, Hugh's going to ask. I know he's going to ask. We'll get there. Okay, we've got another slide, another couple of slides about the urea. Okay, urea is another solute that we play with, and we'll see. Okay. Now, I've included this little link. There's a video, but primarily this video just shows the opposite direction of movement of your vasa rectae blood and the loop. So if that helps you find, that link is there. You can find it on YouTube. Just put in countercurrent multiplier, and there's several videos there for that. So we've got the pumping the salt to get the water out, okay? Now we've got to get it into the blood to finish the reabsorption.
The
or some salt, but mostly the water. So in this illustration, we've got our loop on the outside now, the yellow. See 300 milliosmol. We go to 1200, back up to 1. Here we can see our sodium pumps. It says plus chloride because that's passively following. Here we've got the water coming out of our descending limb of the loop, which we talked about. Here is our vasa rectae, and they put it on the inside of this particular illustration. Okay? But the good thing is they've got the directions going in the right way. Because see, the ascending limb is up. This part is going down. This is the descending limb or our arterial rectae. Notice 300 milliosmol. That's how our blood is starting. That's normally your blood osmolality in your vessels. But as the blood goes down into the medulla, what's happening to the blood? It's getting hypertonic. And as we're going down, we're reabsorbing sodium chloride. A little bit of water is leaving, but not a ton. Mostly we're picking up the salt that was in the interstitium. So there, with the descending limb of the vena rectae, we finish reabsorbing the salt. Now that we've got a hypertonic blood, and this is moving pretty quickly, we're not going to damage our red blood cells, you know, because we talked about crenate and lysis when we put red blood cells in these different solutions. This is going pretty quickly. So when they make this bend and start back up, water is coming in to this hypertonic space because the blood is a little more hypertonic. You see how 900 is low at this part, but 900 is a little bit higher here. So the blood's a little bit more hypertonic than the environment. So water is going to say, you know what, I got a better deal in here. So the water piles back into the ascending limb of the vasa rectae. Boom, we have reabsorbed the sodium, chloride, and now the water. This part of water coming into the blood vessel, the vasa rectae, that's called the countercurrent exchange. Why they wanted the multiplier to be the loop in the water, getting it out, and then the exchange being getting it out of the interstitium to the blood. I didn't set that up, but that's how they describe it. So it's a two-step process of resorbing the water and the sodium chloride in the loop. The countercurrent multiplier, getting it out of the loop and into the interstitial spaces. The countercurrent exchange, getting it from the interstitial spaces into the blood, the vasa rectae. Okay? That's kind of the heavy lifting of moving water and this moving aspect. Here's another way to look at it. We've separated sort of the multiplier out here and our exchanger over here, but you understand they're happening right on top of each other and all mashed together with all the millions of other vasa rectae and loops of Henle. Okay? This again is just the exploded view of what we're looking at. Now we're going to get to this as well in a bit. When we talk about ADH and the aquaporins, that's going to happen in our collecting tubule. And here we have these we don't have the aquaporins in our normal collecting tubule, all right? So here's our medulla getting more and more hypertonic on the left. And if we have no aquaporins, we're completely hydrated, so we don't need to resorb more water. Well, we're going to have really dilute urine at 100 milliosmol that we get out of our body. But if we start to get dehydrated, we're going to open up our aquaporins so this water can come out. And as this water comes out, then it's going to follow that concentration gradient of the medulla. It moves into the interstitium of that hypertonic environment, just like the loop did. And again, we've got some more of these vasa rectae coming up by our collecting tubules. And guess what? The water's going to come out, and it's going to go into our bloodstream, reabsorb that. That's that last hormonal 10% that we can regulate. And so we're going to end up with very, very hypertonic urine because we've got to conserve the water if we're dehydrated.
After collecting, ducts can our bodies do any more reabsorption of water?
Not much. We've done all of the work we're going to do after the collecting tubules primarily.
Okay.
By the time we get to the ducts, it's pretty much been modified all we're going to do.
So in the ureters and in the bladder, there's
No, none. But I think you can envision if you're dehydrated and we're concentrating everything that's in our urine, what happens if you have a super concentrated solution? What could start happening? You did this experiment in elementary school. Crystals can form, and if you have crystallization in your kidneys, what have you got? Kidney stones. And they're not smooth, they're jagged.
So is that solely based on like hydration?
Yes, well, not solely, but primarily. Primarily. And how your kidneys, can function with that. Okay. I know our head's kind of spinning a little bit with that, but hopefully there's enough in there that when you look at it, you go, okay, opposite directions, salt here, water here, and we're good.
Sure, that's all we need to know.
Is that all you need to know? Ish.
That's like.
You know better, right? All right, so moving on, we talked about the complete reabsorption of glucose in the proximal convoluted tubule. We talked about resorbing a lot of stuff regulated and then just eliminating stuff. Here, we're going to talk about resorption of the bicarbonate ion and the hydrogen ion. When usually you hear hydrogen ion and bicarbonate ion, what should you immediately be thinking about in your mind? Chloride shift. When you think hydrogen ions, hydrogen ions are going to do what to a fluid? Acids and bases. So here, this resorption of hydrogen ions and or bicarbonate ions, we're talking about balancing your pH. Because is the pH of your blood important? Yes. You don't want it too high, you don't want it too high, and that range is very narrow. Just like your sugar, you don't want too much, too little. You don't need too much, too little potassium, calcium, a lot of stuff you got to regulate. pH is one of them, and we do that by moving or reabsorbing more or less hydrogen ions or bicarbonate ions. So our hydrogen ions, we're going to filter some of that, but most of those are going to stay in the blood. Most of those are kind of associated with our red blood cells, that process the carbon dioxide. All right. Bicarbonate. Where do we transport carbon dioxide in the blood? That's kind of a trick question. In what form? The bicarbonate ion. That's our carbon dioxide transport. So most of that's going to be filtered. So if we filter the bicarbonate that's working as a buffer against the hydrogen that's inside the red blood cells and in the blood, that's going to be bad because our blood's going to get too acidic. So we have to reabsorb the bicarbonate. And so here we're going to resorb, 80 to 90% of the bicarbonate in the proximal convoluted tubule. The loop is going to resorb 10 to 20%, just depending on how much you filter. And even the distal convoluted tubule can do that as well. We're going to get that bicarb back. Your urine's going to principally be acidic. It's going to be on the acidic side. Now, here in the distal convoluted tubule, we can see for sodium, okay, we're pumping sodium out of our filtrate, right? Sodium potassium pumps. In this case, we're not looking at chloride. Chloride's passively going to follow. But in this sense, to balance the sodium out and reabsorbing, either calcium or hydrogen, those protons, are going to be put back in. What's going to determine if it's potassium or hydrogen?
whichever you have more of.
Whichever one you need to keep or whichever one you need to eliminate. You can balance with either one. Now, in certain situations, you might have a problem. I really don't want to balance either one. You're going to have to pick because we've got to keep the sodium. Sodium's coming out to help us with water transport. So that's the balancing act that we do. So If our blood's getting too acidic, we have some of these cells that are going to be able to reabsorb the bicarb, and they're going to excrete, put into our filtrate, that secretion. That is going to increase our blood pH because of the bicarb and getting this out of the blood. But you can see putting that into the filtrate, it's going to become more acidic, right? If our blood's getting too high in pH, the opposite's going to happen. We're going to secrete bicarb and reabsorb the protons. So again, it's a balancing act. The protons and the bicarb, it's almost like sodium and potassium. They're opposite, whichever one you have to use. Now, do we have sodium bicarb, or do we have bicarb ion pumps? No. How did we make bicarb in the blood? That enzyme? Carbonic anhydrase. Guess what we've got in our loops? If we look in the proximal convoluted tubule where we reabsorb, what, almost 80% of our bicarb, on the surface of our PCT cells, we have carbonic anhydrase in the membrane. So that is going to take the bicarb, we've got hydrogen ions, carbonic acid, carbonic anhydrase is going to make what?
Water and CO2.
CO2. CO2 is going to follow its concentration gradient, which is going to be into the cell. And what do we have inside the cell? Carbonic anhydrase. Carbonic anhydrase is going to say, you know what, let's turn it back into bicarb. And you remember in red blood cells we had band 3 that could pump it up the chloride shift. I'm not exactly sure the name of this transporter, but we're going to take the bicarb out of the cells and get it into the blood. So we're still using carbonic anhydrase, but in this case, we're not trying to fool with the CO2. We're just using CO2 to basically get bicarb into the cell, and then we can transport it out. So again, a lot of these same mechanisms we can apply to a whole lot of different systems. Like calcium is used everywhere, and sodium potassium pump is practically everywhere. Here, we're reusing carbonic anhydrase, but in this case, we're using it for the purposes of bicarb movement, not so much for the CO2. All right, urea. We finally got to the urea. Urea is a breakdown product from proteins. It's nitrogenous. So we have amino acids that have nitrogens as a part of them. And so what we're going to see with urea is this is filtered. And where does filtration happen? The only place it happens? Capillaries, we call the glomerulus. The glomerular filtrate, so it's going to be filtered. It's going to be reabsorbed, and it can be secreted, moved out of the filtrate and back into the blood. The amount that you process is largely going to depend if you're on the keto diet. No, if you're just eating a lot of protein. If you eat a lot of protein, you're going to make more urea because you got more protein in the system and in your blood. 50% is filtered. The remainder is going to be secreted into your urine.
So secretion goes both ways. It goes from the tubule into the blood and from the blood into the tubule.
So filtration is out of the blood into Bowman space in the renal corpuscle.
Yes, sir.
Reabsorption is out of the tubules back into the blood. Secretion is out of the peritubular capillaries or the vasa rectae into the tubules. Well, out of the blood into the tubules. But since it's not the renal corpuscle, we don't say filtration. We call it secretion. We're reserving filtration just for the renal corpuscle.
Is there any way that you could dumb that down just a little bit?
Which part? Now, there is that slide that shows filtration over here with the renal corpuscle and the arrow, reabsorption, moving from the tubules, the filtrate, back into the peritubular capillaries, the vasa rectae, and then there's, I think it's green, maybe, secretion. Materials are being transported specifically out of the blood from the peritubular capillaries, vasa rectae, back into the urine. Slide 39. The glomerulus, it's non-selective. Almost everything's coming out. But with secretion, and some of these use excretion, it's selective just for certain things that we've got to eliminate from the body. All right?
It's not both ways, it's specifically from the blood into the tubular.
That's secretion. Yeah. It kind of sounds like filtration, but since they're in different places, and we're talking about highly regulated secretion versus everything coming out with filtration, that's why we use the two different terms.
For bicarbonate, does it not happen in the distal and the collecting tubule?
It can, but not to a degree, because you're getting almost 100% out from the proximal and the loop of Henle.
Okay, so it's like almost none.
Correct. Most is coming out because the important stuff like glucose, we immediately get out. And we're trying to balance that out as well.
And just to be for sure clear, can you consider the peritubular capillaries and the vasa rectae to be the same thing?
Yes, yes. They're just different. The vasa rectae are the straight ones. The peritubulars are just that spaghetti all over. Yeah. All right. So now these extra pictures, they're going to be in the video that I'll make if my audio is working here. Otherwise, I'll have to make one tomorrow sometime. So here, again, we've got our loop. They're trying to say, okay, this would be proximal convoluted tubule. Green is sodium chloride, the pink is urea, and blue is water. So this is our descending limb where water comes out. This is the ascending limb where we're pumping out sodium and chloride, okay? And when we get over here to our distal tubule and down into our collecting tubule, I like tubule, not duct here, tubule, this orangish pink arrow is showing the movement of urea from the duct, tubule, out into the interstitial space. So that's reabsorption, right, of urea. Urea being a solute, that's going to contribute to the interstitium becoming more hypertonic. But for the case of urea, over here in the ascending limb, urea is going to get brought back into our loop of Henle. And in this case, secreted from the interstitium back into the ascending limb of the tubule. And that's going to add ions to that filtrate while we're reabsorbing the sodium and chloride to make sure we're maintaining the tonicity of the interstitium and that filtrate. And what you essentially see is urea is being brought in here. It's going to cycle back around, and guess what? You see this cycle of urea? It's going to make this loop back and forth and back and forth. So urea is filtered, reabsorbed, and secreted. All three is happening. And so you're never going to clear all the urea from your blood. You're not going to clear it all from the kidney, and some's always going to be in the interstitium. It's helping with the tonicity while you're eliminating as much as you need from the nitrogenous waste. Okay? Now we also have uric acid. This is another nitrogenous waste product. We're talking about nucleic acids here because we have nitrogens as part of our nucleic acids. We're going to break them down in the liver with the various enzymes we have in the liver. So uric acid, filtered, reabsorbed and secreted, sounds a lot like urea. Again, this nitrogenous waste stuff. 90% reabsorbed after it's filtered in the proximal convoluted tubule. And then lastly, creatine. You ever heard of creatine?
Yes, sir.
We heard that in muscle, right?
Yes, sir.
Creatine phosphate. Those proteins break down. We have muscle cells that get damaged. Maybe you're taking supplements, the creatine phosphate. A lot of those supplements instead of going into the muscle, they're going into your toilet, so you're spending a lot of money. Because we're going to have the creatine, it's filtered and secreted. Creatine is filtered and it's secreted. So if creatine gets in your filtrate, it's going into the urine. You're not reabsorbing any creatine.
So how do you reabsorb?
Are we talking,
You don't.
Are we talking about creatine or creatinine?
I'm sorry. Creatinine. So this is a breakdown product of creatine phosphate. It's not exactly creatine phosphate. So do you understand filtered? Secreted means we're eliminating it. And with no reabsorption, we don't want to keep that. It's a waste product we don't want to fool with. Okay?
So there's no regulation of creatinine?
Nope. It's filtered. Well, the regulation would be secreted. If there's too much, you're going to secrete it to make sure you get rid of everything.
Well, how do they come up with normal numbers then on a urine test?
Whew, that's a good question. You shouldn't have, well, they're going to compare the blood level of creatinine with your urine. And they have a lot of different factors that they compare with like that, especially to get at glomerular filtration rate. Yeah.
For the creatinine that's not secreted, what does it do in your body?
Well, it's going to circulate in your blood, but every time it comes through the kidney, if it gets filtered, it's going to be in urine. Not all the creatinine is going to be filtered if it comes through the kidney. There's some substances that have to make a couple of passes through the kidney before it's eliminated. Creatinine is going to be one of those that if it just gets through without happen to fall into that exit ramp in the loops of the glomerulus, then it might make it the next pass, it might not.
And it's just a byproduct?
Yeah, we're not using it, it's a waste product. Yeah, we don't want to keep it. That's why we don't reabsorb it. All right, so we finally made it out of the kidney. We got through our collecting duct, collecting tubule, minor calyx, major calyx, renal pelvis, into the ureter, and we're urine. Water, we've got some solutes, the sodium, the chloride, the potassium. If you got glucose, ketones, proteins, especially if you got erythrocytes and leukocytes, something's wrong. The kidney's not working well, you've got some kind of infection. These are all diagnostic things in the urine. And there's our 1 to 2 liters. 2 liters, you're pretty busy running to the bathroom. But have you seen the people that carry around the gallon jug of water? Yeah, you drink a lot of water. I tell people you lose weight when you drink a gallon of water because you're running so many times to the bathroom. That's how you lose weight. But you understand, right? Water is a diuretic. What is a diuretic? Something that makes you pee more. You drink more water, you make more pee, water's a diuretic.
Just saying water's wet.
The water is wet. All right, so the pH of your urine, it's going to be on the acidic side or it can be on the alkaline side depending on the balancing act of your blood and the needs to eliminate from your body. Typically, it's more acidic if you eat a lot of protein, less acidic if you're sort of a vegan. Not that these ranges are dangerous by any stretch of the imagination. We talked about color last time, right? The poster in the locker rooms. Well, this gives you an example. Kind of looks like a swimming pool test strip. You ever done that before in your pool? Urine should be this color. If you're getting to be bright yellow, you're dehydrated. If you're getting down into the dark brown, see a doctor. Drink some water while you're going to the doctor, but see a doctor because you are severely dehydrated. And what gives the yellow color here?
Urea.
The yellow color comes from the breakdown of hemoglobin, specifically the heme group, and that product of the heme breakdown is urobilin. And if that product collects in your skin, what color is your skin going to be? And you call that jaundice. Well, bilirubin, but you call it jaundice, yeah.
Is it bad if it's completely clear? Like I
No, if your urine is completely clear, you're well hydrated. Yeah, no, you're well hydrated. You want it to be that pale. Okay, hang on a second.
Can I tell you a story how I almost died because I thought, or like how I thought I was dying?
How did you think you were dying?
Okay, so my pee's clear, right, all the time.
Okay.
But then one time I went to the bathroom and I had just taken this pill,
Oh, yeah.
a vitamin B complex, and I went and it was neon yellow. I thought I was dying, like really dying.
Anybody had blue urine? There's some pills that will turn your urine green or blue. So, yeah, I don't know what the medications are, but yes, that can definitely influence it. Okay, now I'm going to need help on this next one because I don't have a sense of smell. I was born without a sense of smell. I'm nose blind. It's called anosmia. But after I've seen people that can smell react to cat and dog and baby urine, I know there's something called urinoid. And if you're a parent and your kids are constantly wetting their diapers, you're annoyed all the time. You see what I did? Okay. Now, any cat owners? Litter box. If you leave that litter box unscooped for a while, are you going to be annoyed? Ammonia, that's the smell that you're going to get after it's sat for a while. Ammonia is another nitrogenous product. And so all that nitrogenous material, it can become ammonia and it gives it apparently a pretty distinctive smell. Is that good distinctive smell?
Yes.
The way that I learned I didn't have a sense of smell, my dad was cleaning the bathroom with a cleaner that had a lot of ammonia in it, and he was standing in the hallway because of the fumes. Well, I went in and brushed my teeth. No, I brushed my teeth, I came out, and my dad's jaw is on the floor. He goes, "How did you stay in there?" I'm like, "What are you talking about?" "It's my bathroom." He goes, "No, you couldn't smell that?" I said, "Smell what?" He said, "The cleaner." I said, "No, I couldn't smell it. My eyes were burning, but I couldn't."
My eyes were burning. It's crazy.
So,
So
yeah,
do you not taste very well?
I don't know how to compare
I know.
to everybody else. Now, COVID, when I got COVID, I lost what I could taste, and I just stopped eating because it was awful. So I do have a sense of taste. I don't think my palate is as discriminating as people that can smell because they're tied together. But I think if I could smell, I'd probably be 500 pounds because I would probably, as much as I like to eat now. All right, let's move on down the track. Ureters. What do ureters connect? Kidneys to the bladder. Now, you're going to have muscle in these because the muscle is going to give somewhat peristaltic contractions because you got to move the urine through the tube and into the bladder. So, you're familiar with some of these terms: mucosa, muscularis, adventitia, the same sort of plan that we have for the digestive system. This, the lumen of the ureters, this is typically where you're going to experience the kidney stones. Apparently it's not fun. We've already talked about that before, but they get stuck and they scrape along inside the mucosa of your ureters. It can cause bleeding. Yes, sometimes when you have blood in your urine, it's an infection. Sometimes it's a stone that's scraping, coming down. All right, make it to the bladder. The bladder is our storage reservoir for urine. And when you look at the bladder, it's not really a triangle, but can you kind of imagine a triangle here? Where the top is kind of dome-shaped, kind of like the fundus of your kidney. But if you could look inside the bladder, you do see a triangle, almost like a funnel, that filters down to the beginning of the urethra. That part of the bladder is called the trigone. It's kind of connective tissue, and it's not really going to move Now, we do have muscle in the bladder. The bladder's got to contract like a bulb to squeeze out the urine, so you're going to have that muscle layer in there as well. And when we look at the wall of the bladder, what we're going to see is the muscularis layer. It's going to be 3 layers. We're not so worried about their orientation because you've got this 3-dimensional structure. But all of these muscles, these 3 layers of muscles that do go in different orientations, collectively they're called the detrusor muscle. Because it's kind of like the heart. The heart contracts 3-dimensionally to push the blood out. The detrusor is going to contract 3-dimensionally to squeeze out the urine. And at the base, we have the internal urethral sphincter. And this muscle, including the internal sphincter, is smooth muscle. So what does that mean as far as nervous system control? Involuntary. These are involuntary. Your bladder gets too full, you're going to have a reflexive action. That internal sphincter is going to open up. Now, we do have this external sphincter down here with the urethra. That's going to be the one we have to learn to control. So, this passageway leading from the bladder to the outside of the body, we already talked about this internal sphincter. That's kind of the urethral. It's right at the base of the trigone of the bladder, part of that detrusor muscle. So you don't control it. But the external sphincter that we see right here, this is going to be skeletal muscle. This is controlled by your somatic, not autonomic. Soma is body. So this is voluntarily controlled. Now, you learn to subconsciously control it, to resist the urgency at times. And the older you get, the more that urgency is going to bother you. But this is the one that potty training. You will, another one of these urinoid things about urine, potty training can do that. Some people will say boys are easier. Some will say girls are easier. Start praying right now, your kid's going to get it. Because if they don't, you're annoyed. Okay, yeah. Now, we have differences in the urethra, and we'll talk about this a little more in our reproductive section. But this illustration is largely the female urethra. It's fairly short. The urethral orifice that's in the vestibule of the female genitalia, that is really short. And this portion of the urethra that passes through the skeletal muscle of your perineum, okay, your bottom, that contributes to your external sphincter. But when you look at the male urethra, it's got a little bit further to go. So when you pass through, see, this is the membranous urethra and that external sphincter right here. First, we're going to be surrounded by the prostate, and this is where you have the prostate gland. And the way doctors examine that is through your digestive tract on the back end, the digital rectal exam. Digital, not electronic. Okay? And once we pass the prostatic urethra, the membranous urethra, now we get into what is basically collectively called either the penile urethra or the spongy urethra. That spongy term doesn't mean the urethra itself is squishy, but it is surrounded by this tissue. This is some of the erectile tissue in the penis that is called the corpus spongiosum. So that's why it's either penile urethra, and then you have the external orifice here, or the spongy surrounded by the corpus spongiosum.
When they do a Foley catheter in a male, what happens to the prostate to where sometimes it's really hard to get it through? Like what happens? Because
Have you?
It's like it's just
Yeah, so, well, this is an artist too, right? So, I mean, it's not necessarily going to be a straight shot because this is bent. That urethra is going to kind of go down and curve upward into the body. But typically, the older males get, the larger the prostate gets.
So it gets a smaller hole.
It's kind of constricting on that. And there are various syndromes where that prostate getting too large has to be treated with medications, not prostate cancer so much, but you got to try to do something to decrease the size and that compression on the urethra. So.
No one probably learned about this, but in a nutshell, what does the prostate do? Like, what is it?
The prostate's going to have secretions that help. If I'm not mistaken, you got a couple of glands in there that are going to help reduce the acidity of the pH of your urine to kind of protect the lining of the urethra itself. You're going to have some. Now, here's the other thing about the male urethra. Female urethra, urine only. Male urethra, urine and your sperm. And so the acidity of your urine, 4.5 or 8.0, sperm come into contact, you're going to kill the sperm. So you have these Cowper's glands, you have the prostate gland, and a lot of these are to prepare that passageway so that you're not damaging the sperm as well. And the prostate's one of those. Lubrication of that passageway as well. The urgency and the process of eliminating urine from your body. You would think that would be a simple term, right? No. Micturition. Micturition is the big fancy word for peeing. Eliminating urine and those waste products from the body. And here's two reflexes. You've got the internal sphincter and the external sphincter, smooth muscle and skeletal muscle. So you have the storage and the micturition reflex, and you're not going to have a lot of control with either one of these. Certainly the skeletal muscle you do, but with the smooth muscle, not so much. So the storage reflex, here we got 100 mils in our bladder. We usually make about 1 liter. I don't know if I've ever made 2 liters of urine a day. That would be a lot. So this storage reflex, sympathetic nervous system, we're awake. We're doing our thing throughout the day. We're going to relax the detrusor muscle. When you relax the muscle of the bladder, what's the bladder going to do? It's going to dilate, get bigger. You can accommodate more urine if it relaxes. This is also going to stimulate the internal urethral sphincter. It's going to close. So you can accommodate more urine. You're not going to eliminate more urine. It's going to stay in the bladder.
It stays contracted even when we're sleeping, though, right?
Yes, it does. Now, the external sphincter, we're going to continuously stimulate that. It's the somatic nervous system, but we've trained our brain such that it's going to continuously be contracted, and that's supplied by this nerve called the pudendal nerve, going down here to your perineum to keep that one closed as well. That's the storage. We don't feel the urgency. Okay, right now, if you don't have a need to go to the bathroom, this is in control.
Is that just underdeveloped in children?
Well, they haven't learned that sort of muscle memory, if you will. You know, you go to swing a bat, you're not going, okay, I've got to put the bat here, I've got to move this. No, you've just done it so many times you can do it. No matter how many times I hit a golf ball, I have to think about it, because there's no telling where that's going to go. But here for the micturition reflex, this is when we got to go. We got baroreceptors. They're going to say, you know what, we're getting kind of full. We probably need to do something sooner than later because we don't want to get any more fluid in here. Well, we're going to get signals that are going to our cerebrum. We're also going to get signals that are sent down to the pons. Man, that brainstem with the pons and the medulla, man, breathing, heart rate, and now urination. It's going to help control digestion. It controls so much in the brainstem. But these signals are going to cause that detrusor muscle to start to contract. That's going to put pressure on the urine to get it out. The internal sphincter, this is parasympathetic control because, you know, during sympathetic, fight or flight, we're not worried about that. We're just worried about getting away. Here, okay, we got plenty of time. Let's just chill, go to the bathroom. That's going to relax. And if you have not learned to voluntarily control that external sphincter in the kids, you're going to pee. Babies don't have to learn to pee. And dads, let me go ahead and warn you right now. The women in your life are very much going to enjoy giving you the opportunity to change your first diaper. Let me just warn you right now. You have the new diaper ready. Okay? Boy or girl. Boys, I think, are more dangerous. Maybe they can aim better. I don't know. But either one. When you take that diaper off and that cold air hits parts that weren't cold before, if they haven't finished peeing, they're going to pee all over you. So I got really good about undoing the tape. 3 children, okay? Undoing the tape, having the other one ready. You get that other one on there so the number 2 doesn't hit, and you get that flap over the top that if number 1 comes out, you're saved. Okay? You will impress the women in your life if you do that. Otherwise, yeah, I'm not going to go with the otherwise, okay? Now, conscious control. Again, you learned the feeling. Your brain learns it, right? You don't have to think about blinking. That's skeletal muscle. You don't have to think about breathing. Skeletal muscle. That external sphincter. You don't have to think about holding it or not. But if you got to hold it, you're thinking about it, right? In those long trips, you're patting your foot, you're looking at your GPS. Where's Buc-ee's? Where's Buc-ee's? So you're thinking about it, and that is conscious. Now, if urination doesn't happen with the first reflex, and you add another 200 to 300 mils, your system's going to override your voluntary control. Your bladder's not going to rupture. You're going to wet yourself. So, don't push it. And when you're in the car with the kids and they say, I got to go, find a stop somewhere or just pull off on the side of the road. Because you don't want to be cleaning that out of your car. 500, 600 mils, it's all involuntary at that point.